Your cells are under constant pressure, and so are their mitochondria. When one is damaged, the cell faces a choice: repair it, recycle it, or die trying. Here is the damaged mitochondria. To recycle it, the cell first has to physically dismantle it, splitting off the damaged part, a step called fission. Once the damaged mitochondria is recognized by the autophagy machinery, downstream autophagy factors coalesce to form the phagophore assembly site, and from here mitophagy, the selective degradation of mitochondria, starts. First, a membrane is formed, the phagophore, which expands around the damaged fragment, wraps it completely, and seals it into an autophagosome. That vesicle is then acidified by fusion with the lysosome and its content degraded. The damaged mitochondria is gone. The signals that trigger these are well known. However, what stayed unclear is how fission gets coordinated with this initiation. In a new paper in the Journal of Cell Science, we set out to find that missing link.
To study this, we built a synthetic tethering system. We fused ULK1, the master autophagy kinase, to an anchor on the mitochondrial surface and added a small molecule to recruit it on demand. This is enough to trigger mitophagy.
To find what acts there, we use proximity labeling mass spec, tagging and pulling down the proteins closest to ULK1 at the initiation site. And among the enriched mitochondrial factors, we found a poorly characterized outer membrane protein, MTFR1L. Upon ULK1 tethering, MTFR1L clusters specifically in ULK1 foci, not across the whole mitochondrial network. So how do cells behave without it? To test this, we knock down MTFR1L with siRNA. In those cells, mitochondrial respiration collapses. Cells start dying by apoptosis. Mitophagy flux drops and re-expressing MTFR1L rescues it. So MTFR1L isn't optional, it's required for mitochondrial health and efficient mitophagy.
Interestingly, when mitophagy starts, MTFR1L gets phosphorylated, and that phosphorylation is ULK1 dependent. MTFR1L in turn feeds back to modulate ULK1's activity. Losing MTFR1L affects downstream signaling like ATG14 phosphorylation. And ATG13, a core subunit of the ULK1 complex, becomes unstable, degraded by the proteasome. So MTFR1L also helps to hold the initiation complex together.
MTFR1L sits at the crossroad of mitochondrial fission and autophagy initiation. Lose it and the balance tips towards apoptosis. Keep it and it supports mitophagy. Recruited to ULK1 hubs, promoting fragmentation, stabilizing the machinery and locking into a feedback loop with ULK1 alongside other kinases. In summary, MTFR1L is a key coordinator of mitophagy and a new player in cell survival decision. From Riccardo, Leon and Chris, thanks for watching. You can find the link to the paper in the description.